Gene regulation is the cellular system that decides which genes turn on or off and when. Without it, your cells would behave like a chaotic orchestra with every instrument playing at full volume. This video introduces the major control points in transcription and translation so that you can finally see how cells keep everything under control. Understanding these processes is essential not only for AP and IB exams, but for appreciating why neurons differ from muscle cells despite having the same DNA. We'll compare bacterial and eukaryotic strategies, highlighting the molecular players, and explain why efficiency matters. By the end, you'll see regulation as the elegant traffic control system of the genome, minus the road rage. Let's begin our extremely orderly journey. Transcriptional regulation determines how much RNA is produced from a gene, which is often the most energy efficient time to exert control. Cells accomplish this by influencing RNA polymerase access to the DNA template. Regulatory proteins such as activators help polymerase bind, while repressors push it away like molecular bouncers. In eukaryotes, chromatin structure adds another layer, with histones either tightening their grip or loosening it depending on chemical modifications. This regulation ensures that unnecessary proteins aren't produced when they aren't needed. It also allows cells to respond rapidly to environmental changes. Think of it as managing the opening hours of a store. If nobody needs your product, you simply keep the doors closed. Prokaryotes love efficiency, so they group related genes into operons, like bundling your streaming services, except these bundles actually make sense. The lac operon turns on only when lactose is available, ensuring bacteria don't waste energy on enzymes for lactose digestion unless the sugar is around. Meanwhile, the TRP operon shuts down when tryptophan levels are high, preventing overproduction. Operator sequences act like regulatory sequences or regulatory switches, controlling RNA polymerase's access. This arrangement helps bacteria respond quickly to environmental nutrient changes. It also makes operons a major point of emphasis on biology exams. If you understand the lac operon, you're already halfway to a 5. Eukaryotic regulation is more complicated because eukaryotes enjoy a little drama. Promoters sit right by the gene, but enhancers may be thousands of base pairs away, waiting for activator proteins to bind. DNA looping brings enhancers physically close enough to influence the promoter. This allows fine-tuned control of gene expression with multiple proteins working together in regulatory committees. Because of this complexity, the same gene can be expressed differentially depending on cell type, developmental stage, or environmental cue. This is how your liver and brain can specialize even though they share identical DNA. If operons are simple switches, enhancers are like a dimmer with 87 sensitivity settings. In eukaryotes, DNA accessibility is everything. Chromatin remodeling adjusts how tightly DNA is wrapped around histones, controlling whether genes are readable or locked in storage. Acetylation of histones usually loosens chromatin, making genes more transcriptionally active. Methylation is more complicated. It can silence or activate depending on where it occurs. Nucleosome remodeling complexes slide or remove histones to reveal promoter regions. This layer of regulation allows long-term and cell type specific control. Essentially, chromatin determines whether your genetic library is open and lit or closed and covered in dust. Transcription factors are the molecular decision makers that determine when transcription should begin. Each factor recognizes specific DNA motifs, ensuring that only the correct genes activate in the correct cells. Activators help initiate transcription, while repressors slow or halt it entirely. Most eukaryotic genes require multiple transcription factors working together, creating enormous regulatory flexibility. This is called combinatorial regulation, and it allows organisms to reuse the same factors in different combinations to generate diverse outcomes. Because transcription factors respond to signals, they link environmental conditions to gene activity. They're basically the managers of gene expression, hopefully better than some managers you've known. After transcription, cells still have plenty of opportunities to adjust gene expression. Alternative splicing allows different proteins to be produced from the same gene, expanding protein diversity like a molecular choose your own adventure. The 5' cap and poly A tail determine mRNA stability and translation efficiency. DNA binding proteins influence which mRNAs survive and which are rapidly degraded. MicroRNAs can silence mRNA by base pairing with them and preventing translation. These mechanisms ensure precise protein levels and dynamic responses. Protein or post-translational control allows for fine-tuning similar to adjusting the seasoning in a recipe. You don't always need more ingredients, just subtle modifications. Translation initiation is a major regulatory checkpoint because controlling protein synthesis is both fast and impactful. Cells modify initiation factors to globally increase or decrease translation rates depending on stress or energy levels. Or specific sequence elements in the 5' untranslated region can boost or inhibit ribosome binding. This allows cells to prioritize the production of certain proteins over others. Because translation consumes large amounts of ATP, regulation here conserves energy. Rapid translation control is crucial in processes such as immune responses and early development. It is the equivalent of deciding whether to start cooking now or waiting until guests actually arrive. MicroRNAs are small RNA molecules that regulate gene expression by binding to complementary sequences on target mRNAs. When bound, they typically recruit the RISC complex to silence the mRNA. This can either block translation or trigger degradation entirely. miRNAs regulate a significant portion of the human genome, making them extremely important in development and disease. Their specificity allows nuanced control without needing to shut down entire pathways. Many cancers involve misregulation of mRNAs, highlighting their biological importance. They are tiny but mighty, proof that even the smallest molecules can run the show. Even after translation, proteins may remain inactive until they receive specific chemical modifications. Phosphorylation can turn enzymes on or off quickly, functioning like molecular light switches. Ubiquitination marks proteins for degradation, allowing the cell to remove worn out or harmful proteins. Post-translational regulation gives cells rapid control over protein activity without waiting for new proteins to be synthesized. This responsiveness is vital in signaling pathways, such as hormone responses. The signal ensures protein quality and maintains cellular homeostasis. In short, finishing a protein is just the beginning of its regulatory journey. Gene regulation allows cells to specialize, ensuring the correct proteins are produced for each cell's type's function. It also enables organisms to respond to stress, nutrient availability, and external signals. By preventing excessive protein synthesis, regulation saves energy, a major concern for living systems. Misregulated gene expression contributes to many diseases, including cancer and metabolic disorders. Regulation also ensures developmental processes occur in the correct order and timing. Without these systems, multicellular life would be impossible. So yes, regulation truly matters, and exam writers know it. We've covered the major stages where cells regulate gene expression, from transcription to protein activation. You now understand the differences between prokaryotic simplicity and eukaryotic complexity. You've seen how transcription factors, enhancers, mRNAs, and chromatin structure work together. Regulation is essential for cellular identity, energy conservation, and healthy development. These concepts are heavily tested in AP and IB biology, so mastering them pays off. Each regulatory level builds on the last to create adaptive, efficient gene expression. Congratulations! You now control the molecular dimmer switches of the genome. That's it for now. Don't forget to subscribe, like, and check out Dr. B's tutorials on Amazon for full AP and IB study guides. See the first link in the comment below. For more tutorials and test prep videos, subscribe to our YouTube channel, Dr. B's Tutorials.